Anne,
Yep.
we're going to be presenting Reconstructing the Hydrology of the Mackenzie River Basin preliminary results. So without a further ado, I'll let you take it away, Anne.
Thank you. So my presentation is just going to be about the results from my PhD project, which is the paleohydrological changes in the Mackenzie River Basin, which I presented last time at the student day event. So I'm studying this specifically because in the Western Canadian Arctic, it's subject to Arctic amplification, which means that surface temperatures have been increasing by 2 to 3 degrees Celsius, which is about twice the rate of the global average. So when we're looking at the Mackenzie River Basin, it's located in the west of Canada, spanning the Yukon, Northwest Territories, British Columbia, Alberta, and Saskatchewan. Specifically, we're looking at the northernmost area where it outputs into the Arctic Ocean. So the map right here shows the all the peat cores that were collected. So the ones that are purple are the main cores that we are studying, whereas the green and the red ones are ones that were collected opportunistically throughout the years. So all of these ones that are in the yellow area were collected in the Gwich'in settlement area, which spans the Yukon and Northwest Territories, around 16,000 kilometers squares of land, and approximately 5,000 people live there, with the largest community in Nunavik. So as you can see again, the 4 purple samples are the ones that are the primary material, whereas the 2 green ones were extra. So looking at the 2 green ones, these are the 16 PS cores, which were collected by GSC officers Colleen Fish and Caroline Duchesne. They were collected using a spade and shovel method in 2016. And the bottom 2 figures here are just the sites. And then there's the 2 samples that were outside of the basin, which are the 18 GTA cores, which were collected by GSC officer Jennifer Galloway in 2017. This is an example of one of the peat monoliths that we, not me, but my supervisor collected while doing geological bedrock mapping. So this wasn't collected using a core, but just by a shovel. And then we have the actual Mache cores, which were collected by Sarah Lord and her team at the Gwich'in Renewable Resources Board in 2020. There's 4 sites that were collected, but there's also sites 5 to 7, which were for vegetation and surface moss samples. So these cores were collected using a titanium core, and in the field, the team measured depth to water table, pH, and electrical conductivity. So these are just some photos where, I don't know if you can see over here, but this black area is where the water hits. So depth to water table is just ground surface level to where the water reaches. And this is the titanium core that's being used by the team. And then the cores were basically packaged up, frozen, shipped to the University of Alberta. So at the University of Alberta, my colleague Andrei Alexandrenko measured pH, electrical conductivity, and total dissolved solids. And what's interesting is pH. Only one of the cores has a pH of less than 4, which is Mac S3. So this suggests that only one of them is umbrotrophic. And umbrotrophic bogs mean that the only source of water is from rain. So they perfectly preserve atmospherically deposited metals and fossils that we are going to be using to reconstruct the paleohydrology. So this is an example of the sampling methodology. The peak cores were sliced at 5 millimeter or 1 centimeter intervals. The 16 PS cores were sent to me at Carleton, whereas the rest were sent to Andre at the University of Alberta. So each of the slices were then subsampled for various analyses, including radiometric dating, geochemistry, and paleontological analyses. So just to summarize all the analyses is this slide, which is quite text heavy. But in general, we were sending samples for Hawk to GSC Calgary. ICPMS was at the University of Alberta. Mercury analysis will be going to GSC Atlantic. Stable isotopes at Manchester Metropolitan University. Plant macrofossils were sent to Marius Galtka, palynology to GSC Calgary, and then I'm studying testing amoeba at Carleton with Tim Patterson. So the results that we currently have back are the dates and some of the Hawk pyrolysis results and the plant macrofossil data. So this is the results for the radiocarbon dates. We currently have some that are being processed, but we don't have any returns for those yet. So the oldest core is actually the Mache S3 at 4,993 years before present, and the youngest is 18GTA292 at 214 years before present. So the 292 core mostly returned greater than modern values. And Mache S3 also happens to be the one core that has the most radiocarbon samples submitted at 10 samples, whereas the rest have around 5 to 8 each. So in general, each of these age depth profiles show increasing age with depth, which is expected, though there's some age reversals, but they're within the expected range of error. Here are the results for the plant macrofossil analysis that was conducted by Marius Galtka. So plant macrofossils can be used to reconstruct the climate and ecology throughout time because the core represents, the depth of the core represents time. So for example, we're looking at these green highlighted parts are the mosses, and they are split into zones, and each of these assemblages can characterize different ecological tolerances. So this one's really clear in the sense that the bottom half of Mache S1 is dominated by Sphagnum lindberghi and Sphagnum angustifolium, and then it disappears after a depth of around 23. So when we're looking at Mache S2, the similar thing happens where the top has a lot of scorpidium, and then it disappears. So this defines different zones. And then we're looking at Mache S3, which is the one peatland that is ombrotrophic. You see something very distinct. So that means there is a distinct shift in ecology through time, which I still have to study. And then Mache S4 is a little less obvious, simply because at the bottom here, there weren't that many plant macrofossils. And then when I was preparing them for radiocarbon dates, I had to submit bulk samples because there weren't any plant macrofossils preserved. And then we also have the Hawk pyrolysis results for the GTA cores and the Mache cores. This figure is just for the GTA cores. And we're looking at the S1, S2, S3, and residual carbon signals. So S1 is just the lipids and sucrose. S2 comprises cell wall lipids derived from algae. The S3 fraction, mainly carbohydrates, lignans, and plant standards. And residual carbon is just the terrigenous plant materials. So for example, at the bottom of 18GT79, down here, there's a low amount of of organic matter overall. And this is the results for the Mache cores, which is a little bit messy. But I found for Mac S4, for example, there's also a dip right here, which is reflected in all of the signals, which implies that there's a drop in organic matter at this time interval as well. And then for me personally, now I'm working on identifying testing amoeba and characterizing the species assemblages. So testing amoeba are biological indicators that are sensitive to climatic and ecological change. And because they respond so predictably to water table depth, they're well preserved and they're ubiquitous. They can be easily collected and identified to species level, grouped into assemblages, and then that can represent specific environmental conditions. This is important because we will add this data into a training set, which will be used to develop a transfer function, which will then be used to reconstruct the hydrology of the Mackenzie River Basin. So those are the results, and the next steps is just to quantify the tested amoeba assemblages in each of the cores, of which there are 429 samples, and then hopefully get a transfer function that can help us reconstruct the hydrology. And that's it. Yeah.
Okay, thanks, Anne. We got a few minutes for questions for Anne on her presentation. Any takers? Oh, Gio?
Paul doesn't ask any question. I'm surprised. And how long will you expect to achieve those objectives from now?
From now? Oh man, I hope to get all of the test amoeba counted by the summer. But that also depends on how fast I can take photos and send it to experts and hope that I got them right. So
okay, yeah. So a lot of constraint you don't have control on it. Okay, thanks.
Yeah.